Thank you for joining us at Biodiversity Conference. Glad to see you all here. Our next speaker is a professor of microbial ecology and physiology here at Humboldt State University. And her courses include bacteriology, microbial ecology, and life at the extremes. Her research investigates microbial diversity and ecology in the high-temperature, acidic, geothermal structures of Lassen Volcanic National Park. And she's here with us today to discuss the roles of microbes in planetary biodiversity and ecosystem processes. Please welcome Dr. Patricia Siering.
Thanks, Bobby. Thanks, Bobby Shearer, for arranging this amazing symposium this weekend. And I'm psyched to be here this morning to take you on a journey. And that journey is going to be into the invisible world that's within the visible world that most of you folks think of when you think of biodiversity. And so the title of my talk, 10 Nonillion and counting, I knew that there were about 10 to the 31st viruses on this planet, but I had no idea of whether there was a term for that number. And so thankfully I could Google that, and I found that in fact 10 nonillion refers to 1 times 10 to the 31st. And so imagine that. Think about that many viruses on this planet. And that's an incomprehensible number for most of us. And just that number alone should give us some insights and give us some impetus to realize that we can't neglect the role of microbes when we consider planetary diversity. And so when I began this talk, first of all, I'm not going to talk too much about my research today, but rather I wanted to kind of give you some insights into the invisible. And so I started to think of, well, how many microbes are there on the planet? And the truth is we don't really know, and there's lots of reasons for that. First of all, most habitats are relatively undersampled when it comes to microbes. As most of you probably know, we don't have good species definitions for microbes. We have no idea how much within species variation there is in natural populations. We don't know how much horizontal gene transfer or the movement of genetic information between non-relatives really affects our kind of phenotypic and genetic signatures that we use to identify species. And even furthermore, for bacteria and for protists, our definitions of species are often based on having organisms growing in isolation in the laboratory. And that's just not how microbes want to grow. So what do we know, or what can we estimate? Well, as I indicated, we think there's about 10 to the 31st virus particles on the planet. Most of these are bacterial viruses, so I don't think you need to go home and start freaking out that, oh my God, there's viruses everywhere. We don't have a species definition for viruses per se, but we can look at genetic types, and we know there's about 1 million genetic types of viruses in a kilogram of marine sediment. We know there's about 1,000 types in your gut, for instance. As far as bacteria and archaea go, the prokaryotes, we estimate that there's about 5 nonillion of these, so perhaps I should have entitled my talk 15 nonillion and counting. Very few described species, only on the order of a few thousand, but it's estimated that we've got about 5 or 10 million. Protists are about the most diverse and least understood of eukaryotes on this planet in terms of diversity. And what's true for both protists and fungi is that the number of described species varies widely depending upon what sources you look at. But the estimates of actual diversity are huge, somewhere in the order of millions. So our concept of biodiversity is very scale dependent. And so for the next couple of minutes, I want to take you on a journey of scale, so to speak, and going from the biosphere down to the level of molecules and thinking about diversity at each of these scales. And so we can look at the biosphere as a collection of habitable environments. But usually we're pretty biased in that view because we think of where plants and animals can live. And in reality, microbes have the ability to live everywhere plants and animals do, and then numerous environments in which we don't find plants and animals. So it's really the microbes that define our limits of the biosphere, or the limits of habitability on this planet. And we can focus in on this scale of diversity journey and focus in on California. And we can look at the ecoregions within California that are basically defined by landmass and soil types and climate and vegetation and the biota within those ecoregions. We can focus in on Northern California and look at the coastal forested ecoregion or the mountain forested ecoregion. And basically just north of what we know as the Sierra Nevada forested mountain ecoregion, you find a national park known as Lassen Volcanic National Park. Lassen. And Lassen Volcanic National Park holds the southernmost active volcano in the Cascade Range. This is Mount Lassen. Most of the park is under about 3,000 meters of elevation. Vegetation is largely mixed pine, some white fir, manzanita, gooseberry, that kind of thing. And we can focus in on a specific region within this park known as Boiling Springs Lake, which is the largest hot spring by surface area in North America. And we can look at Boiling Springs Lake and the surrounding forest. We're at about 1,800 meters here, and so we can look at the diversity of life in that forest. And we can find lots of really charismatic macrofauna, the ones that make us go, oh, aren't they beautiful? One of my favorite, little marmots. And we can look at each of these animals as a collection of habitats. Right? So if you look at, for instance, a bear or a dragonfly, these animals have distinct habitats, each of which harbor distinct communities of organisms. And so you're going to find a different ecosystem in the nose of this bear than you're going to find in its mouth, than you're going to find in its ear or on its fur or in its gut. And sometimes these ecosystems cross paths, sometimes they're connected temporally, but usually we think of them as discrete. Organisms have biogeography, whether you're talking about the scale of a dragonfly or whether you're talking about a bear or our entire biosphere. And similarly, we can take any plant in this forest, and here's a lone cedar tree amidst the pines, and you can look at that plant again as a collection of different habitats, where you've got bark, which is going to house a different community than is housed on the needles, than is housed on the cones, and a very different community than is occupied by the rhizosphere. Okay. And so the idea is that our concept of organism needs to change a little bit, and I'm going to drive this home, I hope, to you in the next several minutes. So we can zoom in even further in our kind of landscape tour, and we arrive at Boiling Springs Lake. And this is like I mentioned, a hot spring. It's acidic. It ranges in temperature from about 50 to 90 degrees centigrade. pH is about 2. And I've been working here and directing an NSF-funded project for the past several years. And it's basically called a microbial observatory. And the goal of this project is to basically understand the microbial ecology of this system. And I'm working with a bunch of co-PIs, including Mark Wilson, who's a geneticist here at HSU, a eukaryotic microbiologist, Gordon Wolfe, who's at Chico, and a virologist, Ken Stedman, at Portland State University. Okay, well, we can go down even further and start to look at this landscape surrounding our lake. And you can look at the mud surrounding the lake. And what becomes, I hope, pretty apparent to you is that macro-scale diversity exists just in the muds, just looking at the mud surrounding that lake. And I ask you to scale down a little further and take a particle of that mud or soil. And in a particle, only a couple millimeters across, there is a diversity of chemistry, a diversity of biology, and a diversity of physical structure. And what I'd like you to think about the scale of these lone microbes in a 2-millimeter particle. That's analogous to somebody who's 5'3" in this audience in a square mile or two. Okay, a square mile or two. Think about the diversity of habitats that are faced in a square mile or two. The same is true when you start thinking about the microscale. And we can look at that microscale using microscopes, and we can look into the waters of Boiling Springs Lake, and we can find eukaryotes, we can find diatoms that only grow in acidic, hot environments, we can find amoeba that harbor very large viruses, we can find some photosynthetic algae, we can find viruses, we can find bacteria, and we can find archaea. All right. Well, we're not done yet thinking about how small diversity gets, because you can look in each of these organisms and look at the DNA and the RNA and the proteins and the lipids that are produced by these organisms. And you can begin to put that together to try to understand what is the true diversity. What metabolic processes are possible in this system? What genes are there to drive certain processes? How diverse are these processes? And I don't expect you to read all of this, but this is the culmination of a lot of physical and chemical and biological and genetic data over a number of years to try to understand how this system works. And one of the things that we've realized is that for some processes, in other words, the breakdown of carbon, and we know carbon comes into the system largely, this is where we're dependent on the trees here, all right, so leaf litter drains into the system and is degraded. Bugs fall into the system and are captured and are degraded. So it's really, in this case, the bugs and plants feeding our bacteria. And we look at it and we can realize that there's a huge diversity of genera and species that function in breaking this material down. Okay. Lots of functional redundancy in this system for breaking down carbon. But we can look at other processes like nitrogen fixation, the conversion of atmospheric nitrogen to ammonia, or the conversion of CO2 to organic carbon. And what we see is we find a very non-diverse system. We find one or two pathways and genes that dominate that system based on the selective conditions that we find in this habitat. So I hope that I've at least given you a bit of insight to recognize that diversity goes beyond what we can see. Okay. And if I haven't made that point clear yet, I want to give you another example. And I'm going to show you these two very sexy, charismatic bacteria. All right. They look the same, right? To most of you? I mean, do you see a lot of difference, or is it only a mother would know? All right, they essentially look the same. They're both about the same size. They're undescribed rods. They're just kind of, I hate to say it, but boring looking. But you look at this guy on the left, and this is an organism called Methanopyrus. This is an organism whose optimal growth temperature is 100 degrees centigrade. Okay. It grows at 122 degrees centigrade. And it grows by basically taking hydrogen gas and taking the electrons off hydrogen, dumping them onto CO2 to make methane. So it uses CO2 like we use oxygen to accept electrons, and it uses CO2 as a carbon source. Okay, pretty cool organism. This is an archaea, by the way. Okay, this guy, common soil bacteria, it's bacillus. You find it everywhere. It's all over your skin, dust, everywhere you want to look, you're going to find bacillus. And this is a kind of everyday Joe bacteria. It's one of the common lab rats of microbiology. And yet it has the capacity, when life gets tough, to make what's called an endospore. And an endospore is the most resistant biological structure known. They basically start this programmed change in gene expression to create these spores that we've been able to revive from samples older than 10 million years. So these, long after we kill all the macroorganisms on the planet, these endospores are going to be hanging out. All right. Now, the kind of an interesting fact is the temperature in the laboratory that we use to kill these is about 121 degrees centigrade, i.e. this guy, oops, I just flashed me, this guy can grow at the temperature that we use to kill the most resistant biological structure known. Okay. And what's even weirder is that if you look at how these organisms function, how they make their DNA, how they make their RNA, how they make their proteins, you might not believe it, but you're more closely related to this organism than this organism is to this organism. And that's because we, humans, are a chimera of archaea and bacteria. And many of the fundamental processes in human biology we got from these extremophilic archaea. So I want to talk to you a little bit about how we can think of diversity. If diversity isn't just what something looks like, what is it? What does it mean to have biodiversity? And so I'm going to talk to you a little bit about some of these different aspects of biodiversity. And the first one is genetic. And this is something many of you have seen before. This is a phylogenetic tree of life, which basically shows the genetic diversity of all life forms. And for those of you who don't know, we consider life to be organized into 3 domains. There's the eukarya, archaea, and bacteria. This particular tree was based on a gene that's involved in protein synthesis that's conserved in all organisms. And the first thing that I hope comes out to you is that you aren't even a blip on this tree. Okay, all of the animals are basically identical in terms of this gene sequence. All right. And when you start to look at other genes that are conserved in organisms, while the relative grouping may change a little bit, the story still holds. There's a lot more genetic diversity in prokaryotes and in our microscopic eukaryotes, which are all these guys, the protists, than there are in all of the macroorganisms put together. Okay. And I think one of the things that got me most excited about microbiology, I actually didn't start out in microbiology. I started out in physics, of all things, astrophysics. I wanted to think about how our life beyond Earth, and I think it's kind of funny that I ended up working on extremophiles in biology. But one of the things that really got me excited about microbiology is when I realized they can eat pollutants, and they can grow on polychlorinated biphenyls, and they can grow on phenol, and they can grow on diesel oil. And the physiologic and metabolic diversity inherent in bacteria and archaea is immense. Often eukaryotes have one pathway, for instance, to fix carbon dioxide, one pathway to break down organic carbon. We've got hundreds of these pathways represented in the prokaryotes. And so if we even look at one particular phylum of bacteria, this is called the proteobacteria, and this is probably one of the most environmentally important phyla of the prokaryotes. You're going to find them in almost every soil, sediment, water. Every plant, every animal has lots of proteobacteria in them. And I don't expect you to get all the details of this, but I want to talk a little bit about some of the metabolism. And so this process, chemolithotrophy, refers to the ability of organisms to basically grow on things like reduced sulfur and reduced iron and reduced nitrogen minerals. And when they do that, they fix carbon dioxide. So these chemolithotrophs are going to be our primary producers in lots of habitats. Okay. Some of these other forms of metabolism, there's a type of photosynthesis that lots of bacteria do that's done in anaerobic habitats, and this is going on over in the mudflats right now. Organisms can grow on methyl compounds, methane, or other carbon-1 compounds. They can breathe sulfate, like we breathe oxygen. And you can even take one genus, take Rhodopseudomonas out here, and look at one species of this organism. And this organism has the ability, it's one species, to grow photosynthetically if it wants to. It can grow photoheterotrophically, that is, using sunlight for energy and organic carbon for carbon. It can grow using fermentation and produce hydrogen. And there's lots of research now going on into this organism in terms of hydrogen production for biofuels. It can fix nitrogen. It can grow on chlorinated contaminants or breathe chlorinated contaminants like we breathe oxygen. And yet it's one species, has this incredible diversity that is going to have potentially very important ecosystem consequences. And so, close to my love and close to my home is the fact that habitats aren't just what you guys think of as habitats where plants and animals live. And I'm going to take you through an array of some of the kind of charismatic places that we might find microbes. And so we can find them living in the heat, all right, and this is Grand Prismatic Spring. It's a neutral to alkaline hot spring in Yellowstone National Park. And to give you a sense of the scale here, this little line is a boardwalk that people walk on. So this is a huge hot spring, and by depth, this is actually the largest hot spring in North America. Boiling Springs Lake is larger and, from space, but this one's bigger in terms of how much water it holds. And what you see is that microbes partition themselves in these beautiful colored patterns based on finding the right temperature and pH and chemical conditions conducive to their growth. You can grow not only in hot surface environments, but deep hot environments. And this shows a picture of a hydrothermal vent. And there are vents off the coast of California and southern Oregon. And in fact, what's going on here is now organisms have to deal with pressure. And in fact, there are a number of organisms that have been isolated that from depths of up to 5 miles deep in the deepest part of the ocean, which is the Mariana Trench. And those organisms absolutely require a pressure of several hundred atmospheres in order to grow. So if you take those organisms and you bring them up to surface, they'll just die because it's of the pressure issues. And lots of the organisms here, most of the organisms here are bacteria and archaea, except you find these really cool invertebrates. And this is an invertebrate called Poganophora. It's a tube worm. And basically this tube worm fills up its tissue with a bacterium that oxidizes reduced sulfur that emanates from the vent, and it fixes carbon dioxide. And so basically these symbiotic bacteria are allowing the worm to flourish here. And there's similar scenarios at methane vents with these giant clams that harbor methane-eating bacteria. So we can go, we have hot terrestrial, we've got hot deep. take a look at this hot acid, and I mainly included this picture because this guy is Tom Brock, and he's got, I don't know if you can see it, but he's wearing this lovely suit for sampling, and he is in many ways the father of thermophilic biology, and they show him sitting on this is Sulphur Cauldron, where he's sampling in Yellowstone National Park. It's typically about 90 degrees centigrade, pH 1 or 2, and when I see this picture, I can't help but wonder if he takes these pants and sticks them in the washer, if they'll completely dissolve because of the acids that he's sitting in. But who knows? All right, and so you can live in hot and acid. All right, and obviously that's the environment that I've been working in for several years. You can also go very salty and alkaline. Okay, and this is someplace I hope some of you have gotten a chance to explore. It's Mono Lake in Southern California. High salt, very alkaline environment, and up at the top we have the edge of what's called Lake Natron, which is one of the Great Rift Valley lakes in Africa. And this is often, if you've seen the nature shows with all the penguins in these African Rift Valley lakes, they often hang out, or penguins, I'm sorry, flamingos, they often hang out by these Great Valley Rift lakes. And the red here is due to the fact that this pH 10 salt-saturated system is full of these archaea that produce a protein that's very similar to the rhodopsin in our eyes. And this rhodopsin, like the rhodopsin that we have in our eyes, responds to light. And so these organisms basically absorb light, and when they absorb light, they can create a proton gradient that's used to make ATP. And so they basically will do this when conditions get rough. There's not a lot of nutrients, but we can make some energy. And it turns out a similar protein exists and is ubiquitous in marine systems, and we find it everywhere in marine systems in these low nutrient environments where organisms need a little extra help making a living. Okay. You can also be salty and not so alkaline, and many of you may have seen this if you've flown over or flown into SFO before. These are saltwater evaporation ponds, and those oranges are due to the presence of carotenoids, which help deal with toxic effects of radiation. And we also find these carotenoids in these guys, which I hope you've also seen. And this is what's commonly called snow algae, or Chlamydomonas nivalis. And you'll find this guy, it's a psychrophile, meaning that it grows in the snow. And when the winter snows come, it gets covered up, so it can't photosynthesize anymore. So it forms these beautifully red pigmented spores. And so in the spring, when the snow melts, you'll see these pink fields of snow. And it turns out snow at 0 degrees is nothing, because you can go into the permafrost and around the South Pole and look at snow and ice in the South Pole and find out that organisms can metabolize down to 20 below 0 centigrade. Okay, and these small little brine pockets that surround crystals of ice. And when you look at the DNA of this organism, you find out it's most closely related to this organism, which is known as Deinococcus radiodurans, which is often found growing on the side of nuclear reactors. You can basically hit this organism with about 1,500 times the radiation that will kill you instantaneously. It will shatter the chromosome into thousands of pieces, and yet it's going to keep on going. So I've given you a little bit of insight into the scale of diversity. We've talked a little bit about types of diversity. And now I want to kind of focus a little bit on what's this diversity doing for our planet. Certainly, I'm guessing everybody in this audience is well aware of the fact that microbes are critical in terms of nutrient cycling in ecosystems. And, you know, most of us, well, except for me maybe, most of us really don't think about nitrogen and carbon cycling too much in our daily activities. But let me tell you, if all of a sudden the microbes stopped fixing nitrogen for you and stopped oxidizing those nitrogen species, life as we know it on this planet would come to a halt. All right? We are absolutely dependent in every ecosystem on the planet. We are dependent on these organisms to basically cycle nutrients that plants, that insects don't have the ability to do. There's been, for years we've known the role of bacteria in terms of waste treatment. We in this country are very fortunate. We flush our toilets, don't ever think about it. We go to the water fountain and get a glass of water. But yet, access to clean water is probably one of the world's largest critical problems in this future. And I often tell my students, if you want to make a difference, go into water quality, because there's so many places on Earth that don't have access to something that we so readily take for granted. Certainly biofuels has been a big push in the past 10 years. Microbes are intimately involved in being studied for their role in lipid-based biofuels, hydrogen-based biofuels, converting plant feedstocks to make our kind of ethanol-based biofuels. And so there's a lot of work right now going into how to make this better and more affordable. Lots of bacteria make a type of fat that we can harvest as bacterial plastic. It's biodegradable, and a lot of our biodegradable plastics are based on this polymer. Something I'm very interested in is a technology called microbial fuel cells, which basically harvests the ability of organisms to anaerobically oxidize organic carbon and couple this to basically delivering electrons to an anode, i.e. they make electricity. You can use that electricity to turn light bulbs on. And there's a lot of really interesting research going on right now that interfaces between engineers and physicists and microbiologists to basically look at how to do this efficiently. And lots of folks now are looking at using wastewater treatment systems where the anaerobic part of wastewater treatment is coupled to electricity production. And I think in our lives this is a reality. It's gonna be here before we know it. And since this conference or symposium is on biodiversity, I want to probably spend the rest of my talk today talking a little bit about symbioses and the fact that there isn't an organism on this planet that isn't intimately dependent on the actions of microbes, and that includes other microbes. And what's happened in the past couple years with sequencing technologies is just kind of blowing us away in terms of what we're realizing about the role of plant and animal microbiomes. And you're going to hear about that in a couple of minutes. Okay, so just to make sure we're kind of all on the same page, I want to talk to you about some of what we know about the roles of microbes in symbiotic relationships. And some of this is very new, and some of this is you've heard about for years, and this is the latter. Certainly, most people in this audience, I would guess, know about the roles of fungi, for instance, in helping plants acquire nutrients. Not only do they help organisms acquire nutrients, but they're absolutely fundamental for us to get nutrition out of our diets. If you get the right microbes or the wrong microbes, it can have a lot to do with how much you weigh. All right? The really good microbes that are really good at breaking things down will put more fat on you, whether you like it or not, because they're really getting all of the caloric value in those foods. And lots of microbes are critical for detoxification of environmental pollutants. There's been some crazy stuff that's come out in the past 5 years that, to me, is, I think, one of the most exciting fields of microbiology right now, and it's looking at the roles of bacteria in animal behaviors. And I'm going to give you a couple of examples of this in a little bit, but everything from sexual selection and mate choice to depression and anxiety disorders to kin recognition and social recognition in animals. It's well established that microbes send out signals that we perceive. The bacteria in your intestines are signaling to your brain as you're sitting here in this talk. All right, and these signals have profound consequences at all levels of both plant and animal biology in terms of development, behavior, and health. We know both in plants and animals that the development and functioning of the immune system is absolutely dependent on our symbionts, as well as the ability of certain organisms to tolerate stress in the environment. And I'll give you an example of that in a moment as well. Surprisingly, perhaps it certainly was to me, is that if you don't have the right biota, your gut epithelial cells can't regenerate. You have issues with the size of your cardiac muscle. And those of you who are interested in perhaps more community ecology, there have been a number of really good studies in the past 5 to 10 years that basically look at the role of microbes in structuring plant communities. And whether this also is true in insect communities, it seems to be. There have been some interesting stuff to come out with the honeybee microbiome, and it may give us some insights as to the role of insects in structuring these insect communities as well. And of course, even those we love and know can go bad, and so sometimes these commensals do kind of join the other side and can actually cause diseases. And so I want to give you a couple of examples today. And I picked a plant and an animal and a bacterial example to kind of illustrate some of my points. And this is a tropical pennant grass that you find growing around geothermally heated soils. And the thermometer there shows that this particular plant is growing at about 51 degrees centigrade. And so, obviously, plants don't usually grow at 50 degrees centigrade. And so about 10 years ago, folks became very interested in how this organism can actually grow in these thermally heated soils, and how does it differ from closely related species. And so what they found was not surprising, is they found an endophytic fungus. Okay, and that fungus is called Curvularia protuberata, and this is a cartoon from a review, Nature Reviews in Microbiology on viruses, and this basically shows the grass in a non-colonized state versus a colonized state. Okay, and this work was published in 2002 by Redmond's group. And so it was clear that it wasn't just the fungus, because they could isolate certain of these curvularia and infect a plant and it wouldn't work. So about 5 years ago, they discovered that what allows this thermal tolerance is that, in fact, the fungus is infected with a virus. Okay, so it's a three-way symbiosis, essentially. And they can basically take the virus out of this fungus and then infect the fungus into the plant, and you get this scenario. Okay, you can put the virus back into the fungus, and you get this scenario. And even crazier is they took the fungus with this virus and they infected tomato plants, which I am so not a botanist, so I apologize. But my understanding is this is a monocot, all right? And tomatoes are eudicots, or sometimes called eudicots. And so they could take this virus fungus symbiosis and stick it into a tomato plant, and sure enough, the tomato became thermal tolerant. All right. And so then they looked at the mechanisms for that, and they found it was associated, the virus had genes that allowed the fungus to make a particular sugar called trehalose and to make a pigment named melanin. And they think that the virus brought this new genotype to the fungus and therefore allowed the fungus to now be thermotolerant. And the citations for this work are over there. Okay, this is something I just heard about this summer at a meeting, and I got pretty excited about. Way back in 1976, there was a hypothesis proposed by Gorman called the fermentation hypothesis for group-specific recognition in mammals. And the idea was that mammalian scent glands harbor communities of fermenting bacteria, fermentative bacteria. And fermentative bacteria produce lots of volatile fatty acid, volatile odorants. And so you can imagine that a given community of bacteria is going to produce a given olfactory fingerprint, if you will, because of that different mixtures, different amounts of these volatile organic compounds. And so, as I said, this was originally proposed in 1976. We didn't really know anything about mammalian scent glands except what people could culture in the laboratory. And what's happened in the past 10 years has been this explosion in DNA sequencing ability. And so now we have the ability to actually look at these communities and these different scent glands. And so there is a really cool study. This work is off of Kay Hollecamp, who came here and talked a few years ago. She's an animal behaviorist who's been studying spotted hyenas in Africa for a number of years now. And spotted hyenas participate in this behavior known as pasting, where they basically mark their territory with this kind of fatty, waxy secretion that's, basically sprayed out their anal gland. Okay, and based on behavior studies, Dr. Hollecamp knew that pasting seemed to provide hyenas with information on the sex of the paster, the group, the social group that that paster belonged to, as well as reproductive status, if they were in heat, if they were lactating, etc. And so a PhD student in her lab, Kevin Thies, who did an excellent review, if you're interested in this topic at all, on the role of bacteria in animal behavior, check out this review. It's really good. It's available free online. And what he did is he said, well, let's team up with a microbial ecologist. And so they got this guy, Tom Schmidt, who's an excellent microbial ecologist, also at Michigan State. And they basically looked at the anal glands of female hyenas from lots of different social groups. I think they had 4 different social groups. And they basically did deep community sequencing, where they're looking at all of the organisms present in that sample. And what they found was pretty cool. They basically found that these group-specific, they were just looking at group in this case, that group-specific, these group-specific odors that are perceived as a social group consist of these short-chain fatty acids, and they are strongly correlated. In other words, the olfactory fingerprint was strongly correlated with the microbial community composition in those anal glands. And as good scientists, we know that correlation isn't causation, but what it does provide is some good evidence to kind of test this further. And so now they're kind of pursuing this a little bit. This was just published. I heard it at a symposium this summer before it was published, but a very, very cool story. Okay, so probably not as sexy and cool to most of you, but way more important for the functioning of our planet is what's going on over in anaerobic environments all over the world. So basically, when biomass is broken down, environments go anaerobic very quickly, and that's because the organisms that break down biomass like to use oxygen. So they basically use it up. And if there's no input of oxygen, the environment goes anaerobic. All right. So what's interesting is that in these anaerobic habitats, the breakdown of one, say, fatty acid is often dependent on somebody else removing that product. Okay, and it's a process called syntrophy, which basically means that the concerted action of multiple organisms are required to break down a particular nutrient. And this happens in anaerobic systems. And not a great picture and not as cute as the hyenas, but what I'm showing you here is there's this organism, it's marked SI, and this is a fermentative bacteria that ferments organic acids. In this case, it's fermenting propionate. And it produces hydrogen and CO2 as waste products. All right. But it can't produce it if the hydrogen isn't removed. There's a thermodynamic barrier. Okay, those of you that know chemistry know that sometimes you've got to get rid of the product to keep the reaction going. And in this case, how that's achieved is by this organism, who's a methanogen, who basically is taking the hydrogen that's produced as soon as it's made. And what we're now starting to see in lots of natural environments is that these unicellular bacteria are in fact growing in a multicellular fashion. And these are protein-based connections that are becoming very commonly seen in natural populations. And the exchange is going on via this connection. And some of these protein connections are electrically conductive, and this is one of the ways that bacteria have been used in these fuel cells, is to produce what are called nanowires, where they can basically make a wire that connects to an anode that they can then deliver their electrons to. Okay, and in this case, it's not an electrically conductive protein, but rather just a protein conduit that allows the hydrogen to move. And so I've talked about organisms that are perhaps near and dear to your heart, but I'm guessing you are probably closest to your own heart, and I want you to consider your symbionts for a moment. And for the past couple years, I'm sure you've heard that there are 10 times as many bacterial cells on and in you as there are human cells. We've got about 8,000 species of prokaryotes living on and in us. And this summer, the National Institute of Health published a whole suite of papers on a culmination of a giant study where they were looking at the microbial inhabitants of 250 or so healthy humans. And they basically surveyed 15 separate sites on 240 women, and they surveyed 15 sites on 240 men. Many men and women were sampled on multiple occasions to basically look at how similar are our microbial inhabitants. What factors control the organisms that are in us that are critical to our biology? And this figure is just meant to show you that the diversity is pretty big. The largest diversity by way of taxa or species is in your mouth, okay, and your skin. All right, intestines definitely have the most genes because the bacteria in your intestines are responsible for you breaking down all your nutrients. And so if we start to look at these organisms in you, you got to start to think about the fact that there are a lot more microbial genes in your genome than there are human genes. Okay, and this cartoon is simple, but it's basically made to show you the human genome has about 23,000 genes, but collectively the microbes on or in you contribute about 1 million genes to your biology. Okay, and so as we think about that, you've got to think about, what does it mean to be an individual? All right. And what we think is that for animals, there is no such thing as an individual. Animals are a composite of their microbes. And to think that those microbes don't play fundamental roles in the biology of that animal is just really ignorant at this point in this day and age. But what does it mean? our concepts on evolution are based on selection acting on the individual. But yet here I'm telling you we're not really individuals. We're these composites. And so there's been a lot of work that some of the evolutionary biologists don't like so much, but the microbiologists are really embracing, that is dealing with what our genome really is. And the term for this that is in the literature right now is called the hologenome. And so this is the combination of all of the genes from your microbes and all of your human genes acting together. And this hologenome theory of evolution, I think, is going to profoundly affect the way we think about natural selection and evolution in natural populations. And so this was proposed by Eugene Rosenberg, okay, and his wife, and they have an excellent review that's also available online. And the idea is that the unit of selection on which natural selection acts is the hologenome. It's the host and its associated microorganism. And that the variation that's present within natural populations can be due to changes in the human part or the animal part, or changes in the microbial part of that genome. And so they've done some really nice work, and this was published in these two papers by Sharon et al. That came out in the past couple years. And they were actually looking at this in Drosophila, right? Drosophila, quick mating, big brood sizes, so you can look at evolutionary processes in real time. And what they knew about Drosophila is that the fermentation hypothesis holds, even though it's not a mammal. In other words, sexual selection in Drosophila is based on the perception of hydrocarbons in the cuticle of the fly. And the different hydrocarbons that are produced, the different cuticular hydrocarbons, are what allow a fly to recognize mate versus non-mate. And so they did some really nice work to show that if you rear the flies on different diets, you change their microbiome. And if you change the microbiome, you change the cuticular hydrocarbons, which change the mate choice. In a single generation, they were able to change the mate choice in an animal. Okay, it's a fruit fly, but still, in a single generation. They could then take those flies and put them back on their original media. So in other words, to get their microbiome back. But that change, that mate choice change, persisted for about 37 generations of the flies. So that shows you a couple things. It shows you evolution can be quick. It can be a one-step thing. Okay? And it also shows you that environmental factors are going to affect our microbiome, which will affect, potentially, things about our biology that we may or may not understand. And so I hope in the last 45 minutes or so I've given you a little bit of insights into the invisible world and given you some things to think about. Some of the stuff that I've talked about is on the human microbiome, and anybody who's interested in that, there are immense resources out there through the NIH website. And there's a currently proposed project underway. It's kind of in the forming stages, and it's called the Earth Microbiome Project. And the goal here is to basically survey the world for microbes, to understand basically the metabolic diversity, the genetic diversity, the taxonomic diversity that's present in the planet and what it's doing in terms of ecosystem functions. And so I hope as you leave today and walk around campus and and enjoy the day when you're looking at the beautiful plants or animals or trees, that you take a moment to think about the invisible players that make the biology of those organisms really possible. And if you came here to hear about my research today, I'm sorry I didn't really talk about it today, but I am giving a department seminar in the Department of Geology on November 5th. And anybody who's interested in hearing more either about bacteria or life in boiling acid can contact me directly by email. So thanks for your attention this morning. And I'm happy to take questions.
I don't necessarily have a lot of microbiomes, so.
That's okay.
But I was wondering with the fungi that you mentioned with the panic grass, is that, is there a distinction between fungal viruses, as in if they're considered, because they don't have capsids, or do have sometimes, because there's like viroids, I think they may be called?
Yeah, that's different. This is actually a true virus, so it has a protein coat, a capsid, in other words. Okay. Yeah, it's a true virus.
And is that virus then mutualistic or still parasitic?
That's a great question. And in fact, the review that I got that cartoon from, that is the main argument of the author, is that it's really hard to say whether, you call it mutualistic or parasitic. And it's really kind of splitting words. Lots of viruses have both beneficial and potentially, detrimental effects. But in this case, as far as the fungus goes, it would be beneficial because it allows it to inhabit a new environment that it couldn't inhabit before. So if you think of organisms as kind of wanting to reproduce and get themselves out there, you might think of this as a beneficial interaction.
Right. So I guess it's mostly weighing the consequences more so than a strict defined definition.
Oh, absolutely.
If the fungus, a few of its cells or partitions in its case maybe suffer once in a while from the virus, as opposed to general reproduction of the fungus. And then when you said about other associations, are you referring to like say bacteria that have like prophages that allow them to produce products that like toxins and stuff so they can compete, even though at some point it may emerge and kill the cell?
Yeah.
At some generation level, though, after it's populated.
Yeah, absolutely. Any effect is going to be habitat specific too, right? And so if you have a commensal and it's putting a genetic load on you, in other words, you have to reproduce that organism along with yourself, you could imagine that in certain environments there may be a fitness cost associated with that, but that when you get into the right habitat, that fitness cost is outweighed by the fitness benefit.
Okay.
Yeah. Yeah, and I think that viruses are, we're just starting to understand viruses a little bit more. Obviously, they're even harder to get at than bacteria. But typically what we do is these kind of large-scale sequencing approaches, and we've done this in the hot spring I work in. And what you find is you find all these crazy viruses that have no relationship with anything that we know of. And so almost every new viral diversity study brings up more questions than it answers, because we just don't have a sense of what these all the 10 nonillion ones are actually doing. But yeah, it's going to be an interesting story, I think.
Okay, thank you.
Oh, you're welcome.
I think I have other questions, but I should let other people.
Hi.
Hi.
I was curious about the anaerobic digesters with those protein-based connections that you talked about. And so were all those bacteria different species of bacteria, as they would say? Sharing those? And is that particular to that environment, or do they think that bacteria has those types of connections, like let's say in our body or in other places?
In that particular example, it was a species of archaea was eating the hydrogen, and then a species of bacteria was making the hydrogen. And basically, kind of the paradigm, and I teach a senior level microbiology class here at HSU, and I always kind of tell my students, one guy's waste is another guy's lunch, and that really is the paradigm of microbiology, and that they're the master recyclers. And so these protein connections, the discovery of these were somewhat new. We were kind of surprised by this, and they were first discovered in these microbial fuel cell experimental systems, where we noticed that bacteria were delivering what appeared to be electrons directly to an anode in an artificial battery system. And so then when people started looking, a lot of it is capturing it and not disrupting those interactions when you sample your organisms. And so that's, I think, why we haven't seen them so much, because we take our sample and we mix it in saline and vortex it and spin it around and then make our samples. But the general consensus is that it's probably a really common way for organisms to actually exchange nutrients, is through direct connection. And we know about the fact that bacteria communicate via these small molecules that can diffuse through their membranes. And it's also thought that some of these communication molecules might be delivered through these connections as well.
Are the connections made from the bacteria's genome themselves?
Yeah. And we don't even have a name for them. You know, they're not in any book yet. Nanowires we have a name for, but we don't really have a name for these protein ones. You know, we've long known that one mechanism of gene exchange in bacteria is by what's called conjugation. And basically the two bacteria get very close together, and there's a protein bridge between them that allows DNA to move from one cell to another. And so we've known about that, but these appear to be different. It's not made of the same protein. It's not encoded by a plasmid gene. It's actually in the genome. And so I think we're going to see more of them as we start to kind of try to preserve samples before we look at them.
And then my last question was just, with that use to maybe conduct electricity or biofuels, what would be like any concern of using these things, just how much energy it takes to cultivate them and how much energy you get out, like those type of questions?
I think right now it's purely in the research stage, you know. And one of the things that happens, and the same thing happened with bioremediation and biodegradation. Back in the early 70s, it was discovered that bacteria could grow on lots of the contaminants, say in fuel oil. And so everybody was like, all right, we can engineer these bacteria. They're going to clean up all our toxic waste, blah, blah, blah. And I think what people realized is that we don't understand enough about the basic biology to make that a workable system right now. So right now, most of the studies are fairly small scale. There are some wastewater treatment facilities that have kind of small pilot projects going on to see if we can actually couple, you know, anaerobic waste degradation with electricity generation. It's pretty new. This was just discovered actually by a guy that I went to graduate school with at Cornell, and they had this great paper where they're showing this mud, and it's basically just mud and this little light bulb going on because they had the, you know, based on the metabolism going on to make the electricity. So yeah, so I think it's a pretty exciting field. And like most of the things in microbiology is it really, and I think one of the reasons I like this field is it's really multidisciplinary. You know, you're often dealing with engineers and hydrologists and chemists and soil scientists, and I just find that really, it's an exciting field to be in because you can really ask questions in lots of different ways and at lots of different levels. And so I think it's going to take this generation of students to really embrace that interdisciplinary training and not be afraid of the fact that they don't really get the math and the engineer, that the engineers may have to take, and the engineers may not have to be able to get the metabolic stuff that the biologists like. But together, I think we can make some fruitful collaborations. So I'm hoping, and certainly in my lifetime, to see some of this stuff come to fruition.
Thank you.
Thanks.
Hey.
Hi.
And so on the hologenome, you mentioned that there was about a million bacterial genes compared to the 23,000 human. Has there been work on characterizing protist or virus genes compared to human genes?
Not so much. Most of the work, some virus genes and some protist genes have been characterized in the context of these microbiome studies, but it's mostly prokaryotic in reality. Yeah, and there's actually probably the biggest concentration of viruses in us would be in our intestines, and they'd be the bacterial viruses. And so what you've got to remember, too, is all those bacteria may have integrated viruses. And so when you get the bacteria genome out, you get these viral genes. But as far as I know, nobody's specifically gone after the protists or the viruses.
Okay. And then also archaea in the body?
Yeah, and those million genes includes both bacteria and archaea. Yeah. Archaea play a big role in terms of not as much as you'd think, but they also play important roles in terms of digestion and so on.
Okay, thank you.
I had another question. With the bacillus that you talked about that was really resistant, and then they would find these million-year-old samples and they were still around. I'm not quite understanding what is still around, just its DNA, or is it
No,
still
they can
functioning?
actually revive them. So they've had amber and halite, salt crystals that were millions of years old, 10 to 20 million year old, and they can revive the endospores in there to basically start growing again to active cells. So they were dormant, yeah. So basically this is a mechanism of dormancy that lots of organisms related to this guy use.
Lou, my question is, what is the biggest hurdle behind making a microorganism that could break down glucose from plant stalks that are harvested from the Great Plains to create ethanol directly?
They're actually using a number of different bacteria and archaea to do just that. To basically act on switchgrass is a big feedstock for ethanol production in biofuels. And so there are lots of organisms that produce enzymes called cellulases. And so they're looking for kind of the best cellulase that can function in those situations. But they actually do use bacterial and archaeal biomass to kind of start that process. I don't think personally that plant-based biofuels are our answer. I think that we're trading one set of problems for another, personally. So I'm all for biofuels, I'm all for green, but I think that we've got to think about the fact that, if we're then growing this crop that isn't feeding anybody, just to put in our cars, is that the most efficient way to do it? And the reason this whole corn biofuel thing got going is because we pay our farmers lots of money in this country to grow corn, and that's what we grow, and we had a lot of it. And so the corn-based ethanol became kind of the first, if you will, sanctioned biofuel, because we already had some systems in place to kind of get it going. I don't know what the biofuels of the future will be. There are a lot of lipid-based molecules that both algae and bacteria make that are really low in oxygen, and those are good for fuels. And so if we can find ways to kind of scale this up, that those might be a good alternative. I'm still kind of riding the wave of hydrogen. I hope that hydrogen-fueled cars is ultimately the way we go, but I don't know. It'll be interesting. I think it's going to be in the next 20 years. We're going to have some big changes to our energy.
And my other question is the viruses that affect the fungi. Do you think that maybe in the future there might be engineered viruses to affect other fungi for restoration purposes because of forest migration caused by climate change?
Yeah, people are pretty resistant about genetically engineering organisms and then releasing them into natural habitats. And I think that this first came up with the biodegradation work in the 1970s. And in fact, the scientific community at that time put a big moratorium on recombinant DNA. This was in the early, when we first learned how to cut and piece together DNA again. And so there's a big moratorium to study effects, and then, we realized, okay, it's probably not an issue. But then what we found is that when you put a bacteria that you create in the lab out in nature, it usually just can't compete. The ones in nature are just so much better than it. So I don't know that, I think it's going to take a big kind of social evolution to basically say, oh, you can release these genetically engineered organisms into the environment for restoration type work. So I don't know, whether that's going to happen or not. Is that it? Okay, well, thank you, everybody, and enjoy the rest of the symposium.